Hi guys, welcome back to my channel Shutapa's Biology. Topic is genetics 2. In genetics 1, you have studied different terminologies of genetics and the preliminary or basic ideas about the genetics. And from today's topic, we shall start all the experiments of Mendel. That is the father of the genetics, Gregor Johann Mendel's all experiments, its analytical results, and the laws postulated on those results. Now, you just observe the full video, as in this video I shall give you the question answer series after completion of this video, and you will just try to perform it yourself, but I shall give the answer key also at the last. Okay, so just observe the total video. What I will be giving to the most important questions of the NEET 21-22 session. So we are starting the part genetics 2. Here first we have to know about the Mendelism, as Mendel is the father of genetics and he just started the subject genetics actually. So we have studied about the gene, allele, etc. Now what is Mendelism? We are coming to the term Mendelism. Mendelism has three aspects actually. Number one is Mendel's experiments. All these aspects are forming the Mendelism. That is, number one is Mendel's experiments. Number two, analysis of the result. Of the results, and number three is postulation of laws on the basis of the result, on the basis of the results. So these three aspects are forming the Mendelism. Now after that, first is that the Mendel's experiments. What is Mendel's experiment? Mendel's experiment, firstly he tried all the experiments with all the crossbreeding experiments with the garden pea, and garden pea is, the scientific name of the garden pea is Pisum sativum. Mendel's experiment material was garden pea, and it's the scientific name Pisum sativum. Why he selected this experimental material? Why? Because there are many reasons. Among those, number one is that it has very short lifespan. If the lifespan is short, then within a short period of time, we can observe many generations. That's very important for genetics. As generation after generation, the transmission of the character is the main thing of the genetics. So this thing is very important. And in his experimental material, that is, P has, why the question is like that, the why Mendel selected Pisum sativum. Number one, for short lifespan, for its short lifespan, number 2, easily can cultivate it, number 3, very important thing, many contrasting characters are there. Many contrasting characters like that, many contrasting characters like the flower color is purple and white. The height of the plant is tall and dwarf. Like this, these are the contrasting characters. So many contrasting characters are there. And number 4, flowers are quite large in size to perform cross-breeding experiment. And number 5, flowers are by nature self-pollinating, so performing crossbreeding is easier. So these are the results. For its short lifespan, that is in short lifespan, if the experiment is performed, then we can observe many generations within a short period of time. That is, P is an annual plant. So within 10 years, we can get the 10 generations. And number 2, easily can cultivate it in a very small space in a garden also. So this is the another reason many contrasting characters like that I shall give after this. There are 7 pairs of contrasting characters, and all those are very prominent with the dominant and recessiveness. And number 4 is that the flowers are quite large in size, so crossbreeding performance is very easier. And number 5, flowers are very by nature self-pollinating, so it's easy to perform the crossbreeding experiment with this. Now, the list of the contrasting characters. Number 1, P shape. Dominant and recessive, first dominant and then recessive. The dominant is round and recessive wrinkled. Number 2, P color. Dominant is grey and recessive is yellow. And generally we can get the color green as it's the dominant one. Then pot shape. Constricted and inflated. In constricted we can get the number from outside, how many P grains are there, but in infrared we can't get the number as it's completely smooth from outside. Number 4 is that odd color. Yellow and green. Flower color. Purple and white. Plant size, plant height, tall and dwarf. And number 7 is position of flower. Axial and terminal. Okay, so these are the 7 contrasting characters of P. Now we are coming to the next one, that is the Mendel's first experiment, that is monohybrid cross. Monohybrid cross means the crossbreeding experiments on the basis of single pair of contrasting characters. Okay, so what is monohybrid cross? We can define it as it's the crossbreeding experiment on the basis of single pair of contrasting character. This is the first experiment of Mendel. This is monohybrid cross. The crossbreeding experiment on the basis of single pair of contrasting characters. Okay, so this is monohybrid cross, and I am giving the experiment now. What he suggested or what he taking for this experiment, that is the tall and dwarf plant. So dominant parent is tall plant and the recessive parent is dwarf plant. We are considering. So in monohybrid cross, we are taking the capital T, capital T. This is the pure tall dominant plant. Okay, pure tall dominant, and another is small t, pure dwarf recessive. Mendel took these two plants for the monohybrid cross, and this is the P generation. P means parental generation. In short, it's P generation, and these are forming the gamete capital T, and this is forming the gamete small t. Then in the F1 generation, they are forming capital T and small t. This is the hybrid tall. This is also tall, but hybrid tall, and this is the first filial generation or F1 generation. Okay. Now in F1 generation, we are getting only the tall plants. There is no indication of the dwarf one. But in the F2, if we cross the F1 into F1, F2 is equal to F1 into F1, and that's why in checkerboard we can write it in this manner. This is the sign of male and female. In a crossbreeding, always you have to mention this, otherwise the crossbreeding experiment has no result. It will get 0. So in F2, F1 F1 means capital T, small t and capital T, small t. This will form capital T, capital T, capital T, small t, capital T, small t again, and small t, small t. So in this experiment, we are getting the phenotypic expression is these all are tall. This 1, 2, and 3. These all are tall. So phenotypic ratio is is 3:1, 3 tall and 1 dwarf. So 3:1, and genotypic ratio, that is the genetic constitution, is genotypic ratio 1:2:1. Okay, so this is the experiment, and on the basis of this, the result is like this. Now, postulation of the laws on the basis of this experiment, and this is the first law on the basis of this is law of dominance. First law of Mendel on the basis of this experiment is law of dominance. Law of dominance, and number 2 is law of segregation. Two laws are postulated on the basis of this result. Number 1 is law of dominance, is telling that the dominant characters will be expressed in the F1 generation, and the recessive will express in the F2 generation. And dominant is always suppressing the recessive. So dominant character will be expressed. That's the main thing of the law of dominance. And law of segregation. Law of segregation is that during the formation of gamete, you just look here, that during the formation of gamete, the characters are segregated in this manner. Here, it's not so prominent. Rather, here you can say that it's very prominent. The capital T and small t, that is the hybrid tall, when it will form the gamete, it will form one capital T and one small t. So, during the formation of gamete, the characters are getting segregated from each other, and their paired condition is restored in the next generation after fertilization. Gametes are haploid, so diploid is the organism, and their paired condition is restored, means they are getting diploid in the next generation after fertilization. So this is the statement of the law of segregation. You will get the laws in book, so I am not writing the laws. You will study the law from the book. It's given over there in NCERT book. You will get the law. Now, after this, we are going to another thing, that is it might be new to you, that is the taste cross and back cross. Taste cross and back cross, these are done not during the Mendel's period, but these are just monohybrid cross. And here, first I am discussing about the back cross, then test cross. This is the crossing between F1 hybrid with either of the parent. With either of the parent. That is either dominant or recessive. Crossing between F1 hybrid with either of the parent. Now parents are, if we make the number 1 table or checkerboard, then number 1 is capital T, the dominant parent, and capital T, small t. The product will be like that in the F2. So here 2 are pure tall and 2 are impure tall. So all tall expressions are all tall, same person tall. Number 1 table, and in number 2 table, the crossing with the recessive parent. So small t and small t, and here capital T, small t. So it will get capital T, small t, capital T, small t, small t, small t, and small t, small t. So this is about that 50% tall and 50% dwarf. 50% tall plant and 50% dwarf plant. Now why back cross? Back cross can give, or significance of the back cross, we can say that by back cross we can get the wanted variety in large number. That is, in this cross we are getting all tall, whereas in monohybrid cross we have got 75% tall, and here we are getting the 50% dwarf plant. Now in monohybrid cross we have got 25% dwarf plant. So what's the significance of the back cross? We can say that back cross can increase the number of wanted varieties. So this is about the back cross, and the next one is test cross. Test cross is a part of the back cross, as actually the test cross has this table is the for the test cross. This table for test cross, but I am giving the another table differently. Why I am telling it all. What is test cross first thing? So test cross is the crossing between the organism with F1 organism with the recessive parent only, not the dominant parent, only the recessive parent. But here also I am giving 2 tables. Why? As 2 tables are required to explain it, and what's the need of the test cross, if we say, then it will be clear. So firstly, the definition of the test cross. it's the crossing between F1 organism with recessive parent only. With recessive parent only. So here we shall get the two tables. Why two tables? Number one is with, here it's mentioned that it's the F1 organism. It's not mentioned that it's the F1 hybrid. Actually, we have to give the result or verify the result that F1 is actually the hybrid or the pure plant or the pure organism. Okay, so here also we are giving two tables. Why? I am explaining later again. Capital T, small t, with small t, small t. Result is capital T, small t, small t, small t, capital T, small t, and small t, small t. And so 50% tall and 50% dwarf. And in number two table, if the F1 organism is the dominant, that is the pure plant. If it's pure, then it will be like this. Capital T and small t, small t. Now capital T, small t, that is the result of the monohybrid, solid capital T, small t. Now this is the result of the F1 of monohybrid cross. All are tall, but all are impure tall. All are hybrid tall. So by this experiment, we can verify the genetic constitution or genotype of the F1 organism. If the F1 organism is pure one, then it will give this result. And if it's not pure one, if it's hybrid one, then it will give this result. Okay, so this is the test cross. And why it's test cross? And by the test cross, we can verify the significance of the test crosses. We can verify the genotype of the F1 organism. So this is the significance of the test cross. So now I am going to discuss about the questions. Very attentively, you just observe this and try to solve it yourself, and then you can follow the answers also, which I shall give you later at the last of the video. So attentively just observe, as all the questions are very important for the upcoming NEET, that is of the session 21, 22. Okay. Now the questions are like that. All will be MCQs. Number 1, all MCQs, very important for the NEET 21-22 session. Okay, so observe it very attentively and try to solve it. The first question is, according to Mendelism, which pair of characters is showing dominance? According to Mendelism, which pair of character is showing dominance? Showing dominance. Option A, terminal flower and green seed coat. Option B is that wrinkled sets and green seed coat. Option C, yellow pod and round seeds. Option D is green pod and position of flower. Sorry, axial position of flower. Axial flower. Axial flower. So which pair of character is dominant? Now the next one is, a character which is expressed in a hybrid is called, a character which is expressed in a hybrid is called number 2, a character which is expressed in a hybrid is called, option A, dominant, option B, recessive, option C, codominant, and option D, epistatic. Number 3, Number 3, alleles are all alleles are then the options. Option A, alternative forms of gene. Option B, homologous chromosome. Option C, pair of sex chromosome. Sex chromosome. And option D, none of this. None of this. This is the most problematic one. All of the above and none of this. Then when F1 hybrid is crossed with the dwarf plant, it's a case of giving the options here. Option A is I am giving here. Option A is dihybrid cross. B I am giving here. B is test cross. C is crossing over. And D is reciprocal cross. Okay, so these are the options, and then number 5, I am just rubbing these questions. Number 5 is there. The ultimate biological unit which countable sterility is called genome chromosome. Option A, genome. B, chromosome. C, genotype. And D, gene. So these are the questions from this parts, and now I am going to discuss the answers. I am solving these questions, but you will try to solve this individually by your own effort. Then you will come to understand that whether you have got the complete discussion or not. Okay, but here I am giving you the answer. First question was, according to Mendelism, which pair of characters is showing dominance? The answer will be D, option D. Number one answer is option D. I am giving here the answers. Number one questions D. Then number two. Number two were a character which is expressed. A character which is expressed in a hybrid is called. It will be dominant. So number 2A. So 1D and 2A. I am again writing here by rubbing this prominently. Now I am giving the answer key or solving the MCQs. One, that is, which one is dominant pair? According to Mendelism, which pair of character is showing dominance? That's the option D. That is green cardboard color and axial position of flower. Then number 2, I have given you the list of this contrasting characters where I have mentioned the dominant and recessive things. So therefore we will get this answer. Okay. Now the next is a character which is expressed in a hybrid is called, that is the dominant. As we know that in F1 hybrid we are getting that the all tall plants. So that is the dominant character. Alleles are, in my first video I have told you about the definition of the alleles, and the first sentence of that is alleles are the alternative forms of gene. So the answer of the third question is A. Then number 4, when F1 generation hybrid tall is crossed with the dwarf propellant, it's the test cross. Just now I have given the description of this and tables also, and so this is the test cross and the answer is B. And number 5, that the ultimate biological unit that controls the heredity is gene, that is D. So this is about the all question answers and this part, and my genetics 3 you just keeping to observe it, going to observe it, and then you will get all other questions, answers, all other discussions which are very important for your board exams as well as the competitive exam, that is the NEET. Okay, so just follow my consecutive videos and also attend the question answer series. Bye for today.